Okay. Okay, now it's already 8 o'clock. 8:03, right? Okay, let me see the slide. Okay, so we start our class now. So okay, so today we will finish up the chapter 8 topics about the translocation. So last week we already finished up the transpiration topic, right, in plant. So for today we will cover up the translocation topic, which is the movement of organic nutrient. Last week we talked about explain about the mechanism of transpiration, right, about the root pressure, transpiration pool, that. So it is talking about the transport of water from root to the leaf. So today's topic is about translocation, which is the transportation of organic nutrient from the leaf that produced from photosynthesis, like sucrose molecule, glucose molecule, and other organic nutrient that transport from leaf to whole part of the body. So this is a process we call as translocation. Okay, so normally this topic, okay, so as same as the last week, we know that transpiration process, the key organ that in the plant play the role is xylem, right? Xylem vessel. So for translocation, the key organ is also vascular tissue, and it is under phloem tissue. Phloem tissue will play a role in the key in transport of organic nutrient. So this is the phloem structure, right? So we know that in phloem tissue we have contain 4 types of cell, right? Component cell, and sieve tube cell, and parenchyma cell, and also fiber. But for the cell that take place in the translocation is only companion cell and also sieve tube cell only. The parenchyma cell and also phloem fiber are not take part in the translocation. Only the sieve tube cell and the companion cell take part in the translocation only. So this one just recap. So everyone, you should know that you already learned in Sem 1, know that there are 4 types of phloem cell lah: sieve tube, companion cell, fiber, and parenchyma. So only the sieve tube and companion cell are take part in the translocation process only. Phloem fiber and phloem parenchyma are not take part as well. Okay, so this is the definition of the translocation. So as you see that translocation is the transport of the product from the photosynthesis, mainly is sucrose molecule or glucose molecule, that as from the sugar source to sugar sink by phloem tissue. So from here you need to know what is the meaning of sugar source and what is the meaning of sugar sink. Okay, so if you know that sugar source, you say that it is a source, right? Source meaning that it is an original place where the photosynthesis is synthesized. So normally the sugar source in the plant is leaf lah, because leaf is the places that the photosynthesis will take part, because they contains high concentration of chloroplast compared to other part of the plant. So sugar source is in the leaf, and then sugar sink is that normally is the place that your organic nutrient or your organic solute is utilized, it will be used or will be stored at there. So normally is whole part of the plant lah. So normally in root or in shoot lah, or in fruit. Fruit also is the sink of the plant. Okay, so normally in translocation we have in our syllabus we have 4 hypotheses that you need to learn. First one is about pressure flow hypothesis. Another one is electro-osmosis and cytoplasmic swimming, and lastly is about peristaltic wave. So the most famous one and the most common one that will be used is pressure flow, or another name we call as mass flow. So these two actually are similar, they are same, just different name only. Okay, so we already know that what is the sugar source and sugar sink. So normally this diagram is show you that how the sucrose that already produced in the leaf transported from the phloem in the leaf into the phloem tissue, sieve tube, meaning that it is the process that the sucrose molecule is already produced surrounding in the leaf, any part because we know that in the leaf tissue we have palisade mesophyll and spongy mesophyll, right? And both of them containing chloroplasts, especially palisade mesophyll. And then this part, and then when they contain chloroplasts, they will undergo photosynthesis to produce the organic nutrient, right? So all the organic nutrient later will be transported into the phloem tissue, into the phloem tube, phloem sieve tube. So this diagram show you that how the sucrose molecule is be transported from the mesophyll cell into the leaf phloem tube. So actually, like the water molecule in transpiration process, in water molecule you learn about the how the water molecule is absorbed from the root and transported from the root hair into the root xylem vessel, right? So similar for this is the process that that the nutrient produced from in the leaf and then transported from the leaf into the leaf sieve tube element. So normally it will be all the organic nutrient firstly will be accumulated in the mesophyll cell first, because mesophyll cell is normally located nearly to the sieve tube, into the vascular tissue. So normally when the sucrose molecule is already accumulated in the mesophyll cell, it will be transported through the series of phloem cell. So normally if you see that there are many of mesophyll cell, and then bundle sheet cell, and also phloem parenchyma cell, and finally it will go enter the campanon cell too. Campanon cell. So we know that campanon cell, phloem parenchyma cell, and sieve tube element are the component of the phloem tissue, right? So normally when they are entering, when the sucrose molecule is already entering the parenchyma cell, some of the sucrose molecule will be entered through apoplast pathway. Some molecule, not all, some of them will enter the apoplast pathway, but the mostly pathway that will be used in the sucrose transport from the mesophyll cell into the phloem sieve tube is using simplest pathway lah. So they will finally enter the sieve tube element. So normally in this process, for those that transported into the apoplast pathway, they were using this core transport proton pump that require energy, because if you see this diagram, you will see that the hydrogen ion inside the cell, so normally you will see here is inside the bundle sieve tube element. So the hydrogen ion inside the sieve tube element will pump the hydrogen ion out from the cell into the cell wall of the sieve tube element. So this one will be create the electrochemical gradient already. So this is one type of anti-transport, and then this is the core transport process. Also transport process, why? Because the hydrogen, the pumping of hydrogen ion into the cell wall will cause the electrochemical gradient, so that this now is highly energy, and then it will transport the sucrose molecule together down the concentration gradient, so that the sucrose molecule that accumulated at here will be together with the hydrogen ion pumped into the sieve tube element, the cytoplasm of sieve tube element. So this is one of the example of core transport process that you already learned in semester 1. Okay. So this is the explanation. So almost I already explained all. So normally if this is a very important the sugar movement into the phloem require active transport. So normally compared to the water molecule normally is at passive transport. It is osmosis process, right? But in the translocation process, the sugar normally need to use active transport process. Okay. And then once the sucrose molecule already enter into the cis-tube element, the sucrose molecule will start to transport in the phloem cis-tube element into the sink. So any part lah, like shoot, like root or fruit, all that the cells that require the organic nutrient for metabolism, or the cell that can store the energy, store the nutrient. Okay, so once the already enter the cis-tube element, translocation will be occur. So normally one of the way of the translocation is pressure flow hypothesis. So what is this? So if you see this diagram, this left hand side, so the first step is the step that I already mentioned this at here. So it is the sucrose molecule when your sucrose molecule is already transported into the cis-tube element. Okay, so your first step is to that your sucrose will be actively loaded into the cis-tube element. So this process is you are using require ATP lah, because it is low concentration gradient into high concentration gradient. So this is why that it using the ATP, because it active transport. Okay, so when they are already when the cis-tube element when the sucrose molecule is already enter into the cis-tube, next your sucrose molecule need to be transport into the sink, right? So how can be transport? How it will be transport? So it require the water molecule to help the sucrose molecule able to be transport. You need to know that sucrose molecule is organic nutrient. It is not a solution. You need to know that sucrose is not a solution. It is like a sugar. So if you imagine that if no solution, no water molecule or any liquid flow molecule, sucrose molecule cannot simply directly go from the leaf into the root one. Cannot. So it require a movement. It require transport lah, like it require a car or any transport car to transport the sucrose molecule from the leaf into the sink. Okay, so this one you will require water molecule. So the water molecule out from the xylem vessel. So normally xylem and phloem is located side by side, but the transport process is opposite direction, because we know that xylem transport water from root to shoot, and phloem transport nutrient from leaf to root. Okay, so it is totally opposite direction. So when the water molecule is already when the water molecule in the xylem vessel now is high, we know that water molecule in xylem vessel is high compared to phloem, right? This one is everyone know in logically, because xylem vessel only containing water, so automatically your water molecule in the xylem vessel will be high water potential compared to phloem one. So this one will cause that the water molecule will diffuse out from xylem vessel and flow into the phloem cis-tube. Okay, so this one will increase your pressure, force the pressure to force the fluid pass through from the leaf into the root. Because now your when the water molecule is already come into your cis-tube, meaning that your water potential value now is very high, and then we know that water is moved down the concentration gradient by osmosis process, so that the water molecule now will be transport the sucrose molecule down to the sink. Okay, when the sucrose molecule is already reached the sink, your sucrose molecule will actively transport or passively transport unloaded into the sink. So normally any cell that require the organic nutrient, it will have this unloading process so it can be active transport or passive transport. So it need to depending what kind of cell that the unloading process is occur. But for your syllabus, you don't need to know lah which type of cell is actively transport, which type of cell is passively transport. You just want to know that both transport can be occur inside the sink for unloading process. Okay, so when your sucrose molecule now is already unloaded into the sink, meaning that your water potential value now will be very high now, because all the sucrose, your solute potential now is decreased now, and your water potential will be increased, so that your water molecule for inside the sink for the phloem sieve tube will be diffused back into the xylem vessel again. So this is one type of recycle process lah. So once water is in the xylem, transpiration pull the water to leave again. So you can say that it is one type of recycle process based on the concentration gradient lah. So normally, as I repeat again, when sucrose molecule is already pumped into the phloem molecule, the solute potential now is very high, so that the water molecule from the xylem vessel will be diffused out from xylem vessel and then flow into the phloem tube. And when there are many water molecule already accumulated here, high water potential will force the fluid pass through the sieve plate and then transport the sucrose from the sources to the sink. So when they already enter the sink, the sucrose molecule will be transported into the root cell or any type of the plant cell that require the organic nutrient. And when the sucrose molecule is already entering the sink, so that the solute potential inside the phloem sieve tube now is very low. Is already very low. So conversely, your water potential will be very high. So the water potential will not accumulate here. The water potential will be diffused out from sieve tube element and then diffused back into the xylem vessel. And then this process will be recycled again and again. Okay, so this is the general concept of the pressure flow hypothesis. So normally this is the explanation. Okay, so normally in cold transport involve phloem loading, your sugar will be from a low concentration to high concentration region, right? So normally your sugar will accumulate in the sieve tube element, resulting lower water potential. Yeah, why? Because your sucrose is already accumulated at here. So that when your sucrose molecule is already too accumulated at here, solute potential will increase, right? And then, as I mentioned before, sucrose is not a fluid; it is the sugar, so that it cannot simply transported from the phloem to the sink by facilitated diffusion if there are no fluid at there. So it requires water. So water comes from where? From xylem vessel. So why water from xylem vessel can be diffused from xylem vessel into the phloem sieve tube? Because the water potential inside the sieve tube near the source is very low now. So that the water potential inside the xylem vessel will be transported from the xylem vessel into the phloem sieve tube. So that your hydrostatic pressure now in the sieve tube near the sources is very high, so it will force the phloem sap, your organic nutrient and your sucrose molecule flow along the sieve tube to the sink. So normally at the sink, your sugar will be unloaded by both active and passive transport from the sieve tube element. So when your sugar is lost, your water potential will be increased, so that your water will be diffused back into the xylem vessel again. Okay, so this is overall of the pressure flow hypothesis. So normally another name of this theory is we call as mass flow hypothesis. So both of them actually is same, just that mass flow hypothesis is based on the experiment. Bezier flow hypothesis is we already applied inside the plant. But mass flow hypothesis, we will more focus on the experiment based explanation. So normally this one is very famous when it coming out in objective question lah. So normally objective question, it will come out like this, with this diagram, and then it will need you to give you many statement lah. It will give you many statements, and then you need to identify which statement is true or false based on the mass flow hypothesis, or known as Bezier flow hypothesis. So normally this one concept is similar, but what I say just that it is using the experimental setup to explain the hypothesis. Okay, so normally you see here there are A and B, right? So this is what we call as the permeable membrane, A and B, and then there are the beaker, right? Will contain the beaker containing water. So you need to know that what is A, what is B, and then this one is one of the examples. So maybe it will have a reverse direction one. So the most important is you need to see an arrow lah during your exam. So normally in exam they will give you a arrow, or they will color it. So normally if you see the color, the darker color will meaning that it is more concentrated lah. The lower color one, the light color one, will be less concentrated, more dilute. So if you see this diagram, you know that the water part confirm is xylem vessel, because we already know that only the xylem vessel containing water. Phloem tissue does not containing water one. Not say that does not containing water, does not containing much water like this. Only the xylem vessel containing water much like this. So we know that outside the surface, that this all water molecule, we assume that it is xylem vessel. And then this A is concentrated. You see the darker color, and you see the arrow here. This arrow meaning that the water is diffused into the A, into the A. So what does it mean? It means that A is the source. So in plant, if you apply into the plant, meaning that it is the leaf lah. So why it is a leaf? Because water molecule is diffused into the A. So why water molecule diffused into the A? Because your A now is very highly concentrated. So highly concentrated meaning that there are many organic nutrients inside A. So when there are many organic nutrients inside A, meaning the water potential inside the A is very low. So that the water molecule from the outside this beaker, from the outside beaker, will be diffused into flow into A by osmosis process through this permeable membrane. And then once the water potential is already increased, so that it will transport the nutrient. You see that it will transport the nutrient. You see this arrow. The solution will be flow from A into B. Okay, so when the solution is flow from A into B, so this one is what structure? Phloem sieve tube element, right? So this is the phloem sieve tube element structure, and then this flowing is flow of solution, meaning that our phloem sub is transported from the source to the sink. So when the phloem sub solution organic nutrient already reach the sink, what happened is you see that the organic nutrient now is very low. Because at the journey of the process, your organic nutrient is already diffused out diffused out and become dilute. So when they are become dilute, meaning that your water potential for B is very high. Now, so that the water molecule will be diffused out from B to outside of the cell lah. Outside of the cell. So normally is that the water molecule will diffuse back into the xylem vessel. and then you see this diagram again, so that the water molecule will be transported again from the from the root into the leaf again. So this is the general experimental concept for the mass flow hypothesis. So for this kind of question in examination, normally the first important point is you need to see the arrow first. You need to determine which part is source, which part is sink. So for this one, A is source, B is sink. So it can be the balik one, it can be reverse one. Not necessarily A is left hand side. It can be A at right hand side one. So based on the question lah. So you need, the most important is you can see the arrow, and another one is you can see the color. So the darker color is the source, the lighter color is the sink. So and another one is the flow of the direction inside the water also. So the flow of direction should be, water should be flow from the sink to the source. So you see this kind of direction, you know that B is sink, A is source. Okay. Okay, so far that's all for the pressure flow hypothesis. Okay, next I will explain about the another three type of hypotheses: electro-osmosis, cytoplasmic streaming, and final one is peristaltic wave. Okay, so normally these three type of hypothesis is very cold topic that normally students will not learn it lah, because too boring, maybe too boring, or maybe never come out yet, even in real exam never come out yet in both objective, structure, and even essay. So that's why that one of the reason nobody want to care about that lah. But however, because it is in the syllabus, so that you should know it. Okay, you should know too lah about these three type of hypothesis. Okay, so first is about the electro-osmosis hypothesis. Okay, so when you see this term osmosis, so when you see the osmosis, this process will be using water one. It will including the water. And then if you see the electro, meaning that it will include electrical charge. So that so actually this one is one type of chemistry theory in electro-osmosis lah. But in Stpm syllabus we not learn that. So one of the reason that many people will be confused lah. So anyway, but if you don't know about the theory of electro-osmosis, actually you still can remember it. Okay, so electro-osmosis actually is the movement of ion in an electrical field through fixed powers with its electrical charge by carrying water and potassium ion. So there are three main components that were involved in electro-osmosis. One of the main component is water molecule, and another one of course solute lah, because translocation we need to transport solute, right? So of course solute, and another one is potassium ion. Okay, so how it is become. So normally in your sieve tube and sieve plate and your phloem protein are normally in negatively charged, thus forming a fixed power surface with an electrical charge. So normally in your sieve plate it is in negatively charged one. At your beginning process. Okay, and then when your flow occur through the negatively charged sieve plate, an ion will be repelled, but cation ion will be able to pass through. So you know that this is it at the initial process. Your sieve plate is negatively charged, right? I already mentioned your sieve plate is negatively charged because your protein is negatively charged, so that it will repel any anion molecule. We know that in electrical charge, anion and anion is repelled. Same electrical charge are repelled to each other. So only the cation molecule can be it can only attract cation molecule. So what kind of cation molecule? So in this plant, potassium ion will play the role in cation molecule. Okay, so that your cation molecule, potassium ion will be transported from the component cell into the sieve tube element. So when your mass flow occurs downward through the phloem, the repulsed ion will accumulate above the sieve tube, so that above sieve plate will become negative. And then sieve plate now will be fixed a porous surface within an electrical field, such as is needed for electro-osmosis to occur. Okay, so when there are differences occur across the sieve plate is reached, your proton or hydrogen ion from the wall of upper cell into each cytoplasm will lower the pH value, making the cytoplasm above the sieve tube become positively charged. Okay, so if you see this, so when you want to mass flow occur downward through phloem, your N ion will be accumulate first at the sieve plate first, so that at your initial part, all your ion at here is totally negatively charged at initial process. Okay, so once that within a electrical field, so that it can produce a electrical field. Okay, and then when the critical potential difference is already across the sieve plate is reached, hydrogen ion from the wall of the upper cell into each cytoplasm will lower the pH, and then making the above the sieve tube element become positively charged. So hydrogen ion from the any part of the cell will start to diffuse into the sieve tube element. Okay, so that your charge for the sieve tube element at the above will become positively charged now. Okay, so this increased positively charge push other cation, mainly is potassium ion, by electrical pulsion through the sieve plate from the upper to the lower cell, and therefore your electro-osmosis will be occur. So your potassium ion then secreted on the another side of the sieve plate, and then pressure of potassium ion on the other side of the sieve plate induce negatively charge on the other side of the sieve plate. So it is actually like the macam play the charge between the sieve plate. So normally when the hydrogen ion is already accumulated at here, potassium ion will be accumulated at here, and then this positively charged ion will cause the potassium ion diffuse through the sieve plate. And then when the potassium ion is already passed through, your solute molecule and the water molecule will automatically also pass through, so that the electro-osmosis will be occur. So this shared hydrated potassium ion will carry water molecule and dissolve solute across the sieve tube. So with the energy cost on electro-osmosis, flow of polar water molecule and dissolved solute through the sieve pore to the adjacent sieve tube element. So this is the overall process of the electro-osmosis. So normally it play the charge between the sieve tube element. You will see here it play the charge only. So initially at the beginning, your charge for the sieve tube element is negatively charged. For the upper part is totally negative. And then when the hydrogen ion is already starting to pump into the sieve tube element, potassium ion also will start to pump into the sieve tube element, causing the upper part of the sieve plate become positively charged. Okay, so when the upper part become positively charged, so you see this here. See here. So when the electric charge, so electro-osmosis will be occur. So how it occur? your positive ion on other side of the sieve plate will induce negatively charge on the other side of the sieve plate, creating a potassium difference across the sieve plate, so that your potassium ion will starting to diffuse out, because it need to induce the charge at the lower part of the sieve plate. So when the potassium ion is already passed through the sieve plate, so that the water molecule and your solute will also pass through into it, so that when the potassium ion is already inside the pass through the sieve plate, and then it will be cycled back into the component cell, and then it will be cycled again and again. So it is like based on the electrical charge. So yeah, okay. And another one is about cytoplasmic streaming hypothesis. Okay, so about the cytoplasmic streaming hypothesis, actually it is the flow of the ion of the molecule that can be flow inside the cytoplasm. So normally it had been proposed that your solute can be carried from one end of a sieve tube element to other by streaming, and then transfer across sieve plate by active transport. So streaming is that your cytoplasm or past cell is ordered to move around within the cell. So meaning that actually your organelles or your any nutrient or organelles inside the cell actually can be moved around freely one. So this process is called streaming. And then it found that when you see this diagram lah. Okay, so you see this diagram, any molecule inside the sieve tube element is freely moved one. It is very freely moved. And then the molecule at far of the sieve plate, it will move very fast. But when they are already near the sieve plate, the molecule will move at very slow speed. And then this slow speed will force the molecule at the upper part of sieve tube element, force out from the upper part to lower part by active transport. So molecule will join into the next cycle again. So normally different solute move through the sieve pore at different rate due to the different molecular characteristic. So both energy streaming and transfer the sieve plate will be energy dependent, explaining high turnover of ATP in phloem cell. And then this process is require high energy or ATP lah, compare high energy or ATP, because it actively transport process. And final one is about peristaltic wave. So it is one type of wave that we produce in the plant that transport the solute particle from the source to the sink. So normally this one is that it is using the cytoplasmic filament that found in the phloem sieve tube. So in previously we used about the phloem sac is transported inside the sieve tube anyway. But for this one, peristaltic wave, the sieve tube element, the phloem sac actually is transported between the cytoplasmic filament. Okay, so between this cytoplasmic filament, there will be contract and relax. The cytoplasmic filament can be constrict and relax one, like muscle, like human muscle one. So this one will push the phloem sac from one sieve tube to another sieve tube. And then this process is also require energy, because it is in the opposite direction depending on the metabolic energy and also ATP lah. So it is using the human muscle contraction and relaxation. It contract and relax, like here it contract and push the phloem sac from upper part to the lower part, contract relax until it reach the sink lah. So this one, when it is contract and relax, it will produce the peristaltic wave. So this is the theory about the peristaltic wave hypothesis. Okay, so actually today class is already end. Yeah, today class is end. So I already finished up the translocation topic. So today we just learned about the translocation based on the four hypotheses: pressure flow hypothesis, electro-osmosis hypothesis, cytoplasmic swimming hypothesis, and also the peristaltic wave hypothesis. Okay, so I think that's all for today. So any questions so far? Okay, so if no question, then we will end the class today. So we will see you again next week. So next week I will enter chapter 9 lah, control and regulation. So thank you. So thank you for attending and see you next week. Bye.